And welcome, Mindsetters, to this session of Grade 11 Learn Extra. We're looking at life science today. I'm Ty. I'm here with Lou. Lou, what are you doing today?
We're doing body plans, so
Right.
hopefully they're ready and let's give it a go.
All right. So on that note, while Lou makes his way across the board, Mindsetters, as I always say, make sure you get on the page. You know the link, www.facebook.com/learn extra. Make sure you get on the page and let me know if you're lost in it, if you need help, or if you just want to say hi. Because it's always welcome, and we're always glad to have you on the page with us. But on that note, this is where I'm going to hand over to Luke. Luke, take it away.
Thanks, Tal. Hey guys, well, this section is quite cool. Now, last week we started doing body plans and we had a look at specific things. So what I'm going to do is we're just going to do a quick recap so that you can understand what I'm talking about. But before we start, I've just got a slight problem. The problem is I've been trying as hard as I can to learn these words. Now, these words are big, they're huge, and no matter what I do, sometimes they just don't come out right. So I'm going to try to get them right. Hopefully they come out today. So, Ta, good luck, because I'm going to be asking you as well.
Oh, great. Great.
First thing we're going to have a look at, right, is when we did body plans on last week, Wednesday, right, we had a look at how it is divided. The first thing we did was we had a look how they were divided. You know, you can get certain divisions. For example, you got bilaterally symmetrical, which, of course, if I had a look, if an animal was like that, you could cut it right in half. So half was this side, half was that side. It was bilaterally symmetrical. Now, the next one was radial symmetrical. Right, it's over there. You can cut it in any way you want. It was plain and simple. And then, of course, the last one is asymmetrical, which means you've got no chance on this, in this country or this lifespan, to actually cut this in half. If you have a look at that, you could not cut it in half, right? Now, each one of these things came to a specific phylum. You start looking at certain things, okay? So we look at how in what symmetry they are, right? The next thing we had a look at was if they had a coelom, okay? Which means they were divided into different sections. You got different types of body parts, right? Or different types of body layers, right? You got your single-celled organisms, right? And then you got your things like your diploblastic and your triploblastic, if you can remember. A diploblastic had an ectoderm and an endoderm. And then you got your triploblastic, which had your ectoderm, your mesoderm, and then your endoderm. So just running you through this. So hopefully you're remembering this, right? And when we got to a coelom was inside, right? It was between the endoderm and the ectoderm, right? And it had like a membrane around it, okay? And if it doesn't have that, it's an acoelomate, right? If it doesn't have a coelom, sorry. If it does have a coelom, there can be a false coelom, right? And if you have a false coelom, you don't have, if that is the organism, and then you get this piece over here. If this is a coelom, if you don't have a layer over here from the endoderm, which is normally your gut, to the ectoderm, right? They call that a coelomate. They call that a false coelom. And then you get your normal coelomates, which means it's got a layer on the inside, right? This is just simple. I'm just trying to go over it nice and quickly before we get into the proper animals, right? Next thing we had a look at last week was we had a look at if they had a through gut or not. If you don't have a through gut, you would normally eat. I'll do something like that. You've got, the worst way to put it is your mouth and your anus in the same hole. So you would eat in here, right? Absorb all the nutrients and then leave again out of the same hole. If you had a through gut, you have a, normally like this, you have a mouth and you have an anus. So it goes in there, let's just do that. And this pipe is joined, plain and simple, and the food comes in, gets absorbed, all the nutrients get taken out, and then gets excreted from the bottom, right? That was your through gut. Okay, so now that I've just gone over that very quickly, now we're going to start looking at the animals. Let's start getting into more detail, right? First one we're going to have a look at is your Porphyria. Now, it's a nice big name, right? There it is, Porphyria. These are your sponges. Now, where can I find sponges? right? It's not that difficult. I started last week, I was telling the people that sponges are quite cool. This is where you see these beautiful islands, right? And you go in, you see the sea, it's nice and calm, and you run in and you go look at coral. Now, you know those coral that's there at the bottom, all the different colors? Those are those are normally your sponges. Plain and simple sponges. Now, we need to go into more detail, right? Because these are quite cool. First of all, the first big word we're going to have a look at is sedimentary, right? Sedimentary just means that it's covered with water and it's stuck to the bottom, right? So it's stuck onto the ground. It can't come up for air, right? Stuck on the ground, and it's always underwater. Okay, you can get it in fresh water and in seawater. So you do get them in the fresh water, even though we don't see them as much. They're not as colorful as your seawater, right? Next thing that we have a look at, they are, as I said, they're found in salt water and fresh water. Their body is asymmetrical. Remember? Here we go. Now we start bringing in the other work that I've just come through. Asymmetrical, which means they cannot be cut in any other, that can only be one way. You cannot cut it in half, you cannot cut around it, it's all going to be different. They're not going to be identical. You can't cut it in half, okay? That is asymmetrical. There's that big word. Very nice, okay? It also has 2 body layers. If we have a look at it, 2 body layers, which means it's diploblastic, okay? And if it's diploblastic, it'll have the 2 layers, which is going to be your epidermis, epidermis meaning the outside, and the endodermis meaning the inside, the inner layer, which is your endodermis. Plain, simple, not that difficult, okay? It's actually quite easy, these guys. These guys are not that complicated at all, right? And if we look at it carefully, in between the epidermis and the endodermis, it's got a jelly-like layer, right? It's not cells, it's just like a jelly liquid inside, okay? And it's an important function of it is just to separate the two, but we're going to find out now exactly how this thing eats, okay? And if it eats, it needs that mesoderm in the inside, right, which is a jelly-like substance, okay? So we've got the outer layer containing, this is a nice part. This is a big word. We're getting there. Porocytes, okay? Now, a porocyte, where have you heard pore before? Okay, I know all the ladies. Correct me if I'm wrong. We normally look at the ladies and they always say, oh, haven't got such good skin, or my pores are blocked, or things like that. Pores meaning what? Ty, do you think you understand?
As in, basically the spaces where actually our skin absorbs like air and energy.
So it's something like it. It's not air, but you're getting on the right track. It's holes, small, tiny little holes inside there, right? That is your sporocytes, okay? They allow water in. Now, at the moment, you might not understand what I'm trying to say. I'm going to have a picture just now, and that picture I'm going to explain it all over again, right? So they've got sporocytes, which is quite cool. Then you get your inner layer, which contains conocytes. Okay, there's the next big word. You see, I've been trying hard to get these words right. So the conocytes, right, those things are quite cool because they've actually got flagella. Now, I'm using big words again. There they are, flagella. Okay, flagella is a huge word. Now, I know I'm leaving you to hang. I just want you to remember these words. I'm going to explain it in 2 minutes, right? The flagella do a specific job. They help with certain things, right? And then, of course, your mesophyll. Remember between the endodermis and the epidermis, we had that like that jelly-like substance, right? That's your mesophyll, and it contains that one, amoebocytes. Your amoebocytes, you've got amoebocytes in you at the moment, okay? Amoebocytes does a specific job. Amoebocytes actually go in and they eat things. So they absorb food by eating it. And how do you think a single cell eats food? Okay, please send it to Ty. Let's see if you can give me an answer. How does amoebocytes eat? How does a single cell take in food? I'm trying not to say the word, right? So they take in food in a specific way. Okay, so let me see if I can go any further. There we go. I didn't want to, hopefully it wasn't there. That's what I was checking. There we go. There we're having a look at your sponges or your coral. They're absolutely gorgeous. And if you had a look at them, you cannot cut them. You cannot cut them in half. You cannot cut them in radial sections, right? And if you have a look at it, there's a nice one. Can you cut it in any way to make the one side look identical to the other? Okay, that's asymmetrical. Can you remember this stuff? This is quite easy. You just got to learn the big words and we'll get it right. Okay. Here we go. This is what I was in. Okay, this here, okay, this piece here, this main thing is actually what we're looking at. This is a sponge. Now, if I go back, okay, and I pull this all the way down to go see where I'm just going to have a look at this. You see this red thing here. Hopefully you'll see this nicely. That piece there. Okay, that piece. If I remove that and I took it here in my hand, right, and I'm holding it, okay, and I put it there against the board. There it is. That's what it looks like. There's normally a cover, and there's that piece that I'm talking about. Can you see it? That piece there is that red piece that I was busy holding. Now, the reason, sorry, I just want to get rid of that because that was just to explain something to you. Now, the reason why I put this up here is to explain in detail what we're going through. Okay, first thing we do, okay, this is an organism that doesn't have a coelom. Now remember, the coelom is a jelly-like bag, okay? Now, this doesn't have it, okay? It doesn't feed through the same holes, right? Okay? It doesn't have a through gut, but it doesn't feed through the same holes, which is quite strange. Remember I said to you, if you don't have a through gut, you have this type of thing where it comes in and back out again. Okay, that means it doesn't have a through gut. But this doesn't have a through gut. It brings things through different pores, but it actually doesn't have, it doesn't absorb in a gut. Okay, this is actually where everything happens. Okay, so let's have a look at it. The spore, or the water and the food and everything like that, comes into this huge sponge through a hole. Can you see that I've made, let me see if I can, it's not going to work nicely. Pink. Pink's always working with this thing. If I look over there, I've put a pore, okay? That pore, I'm going to have a look at it over here. It's the same pore over here. Can you see it? I'm just not going to move it around. The water of whatever's around it has got food, it's got water, it's got everything that it needs. That actually comes into this part of the sponge, right? It goes around this area, does what it needs to do, and then leaves out the top again. Okay, the top, very important, osculum. Okay, osculum's a very important part. It's the part right at the top. That's those holes. If you remember on the last slide we had a look at, see these holes over here? That's the osculum, right? They're the ones where the water ends up. Okay, it keeps pushing out. Okay, then let's go back to that following slide. Now, it goes in this pore and it gets thrown out. Okay, very important. But now, how am I going to get the water inside there? That is a big problem. Now remember, I spoke to you about certain big words. I spoke about conocytes. Can you remember? And flagella, right? Remember all those big words. Sorry, I'm just hiding. If I take this endoderm over here, this big piece over here, and I take it out, and I'm actually going to put it down here. That's what it looks like. Okay? Now, watch this carefully. You get a pore cell. Here it is. There's your pore cell, right? So the water's going to come in here. Am I right? There we go. You know how it comes in. But we need to make the water come in. Now, if you're sitting in the bath, right, how do you make the water move, right? If you think about it, you're putting hot water. What do? You want the hot water all over the place. How do you make the water move? You turn it, right? So you notice my hands turning, right? That's what this flagella actually does. Okay? This thing here, this yellow part here, right? Those things actually move, and they create like what's it called? A flow. It creates a flow, and that flow moves the water. Moves the water out the osculum, right? And as that goes out, water gets sucked in to replace it. Okay? This piece here, this cell over here, this whole thing over here, hey, that there is called your conocytes. That cell there, the conocytes, has flagella, there's the flagellum, and that thing moves and it creates the flow and it pulls the water in. Right. Now, not only does it do that, but if I took this piece, or maybe I'm squashing up here a bit, if I take this one here, okay, and I move it all the way down, I'm going to draw it for you so you can actually see what I'm doing. Okay, what actually happens is you have a cell, there it is, okay, and on that cell you have a flagellum, there it is, and then you have this special part. Okay, this special part goes over here. It actually looks, I'm going to try and make it round, actually looks like that, and it comes in like that. Okay, hopefully you can see what I'm trying to draw. Okay, this thing here has a special job. Okay, now what I want to do before we go to a break, I want to tell you this. I want you to go to a break. I want you to go find out what it is. I want you to send me the answer. Okay, I want to know what, if I label it, what this thing is. This thing that I just drew over here. What is it and what is it used for? Okay, so I'll see you after the break. Ta?
All right, so Mindsetters, as Lou said, make sure you get on the page, get those questions in, and don't worry, I'll be posting pictures very soon, so make sure that you guys get on the page. But on that note, we'll see you after this page.